We're going to go ahead and start taking questions from the audience at this point. The first question, one person asked, I noticed precipitation is not listed as a key factor in biological response to climate change. And this person has also heard that there's been a huge increase in precipitation in the Northeast and that the increase of freshwater flowing into estuaries has caused localized declines in pH. I think this question would probably be best addressed by John. Was this factored into your study, and did you still find no significant impact of precipitation on biological response?
So it's not that there's no significant impact on biological response. It's within the framework of the assessment. The projected future increases in precipitation have more uncertainty in them. So some models suggest, very high precipitation. Some models suggest relatively small increases in precipitation. So as we looked at sort of the exposure to precipitation, because of this uncertainty, the exposure of species was lower. But as the person asking the question rightfully, bases their question on, precipitation can have dramatic impacts on individual species or individual systems. And then it is, gets into the idea of coastal acidification, which in our assessment, we were looking at ocean acidification and not coastal acidification. So I don't mean to give the impression that changes in precipitation, changes in currents, are not important. Rather, the dominant factors in the Northeast appear to be ocean acidification and temperature, but those other factors, sea level rise also, will clearly be very important for some species.
Great, thanks, John. The next couple of questions tie really well into this discussion. The first is sea level rise from thermal expansion and added volume from melting accounted for in pH change projections?
I'm not a climate scientist, so that question would probably be better to an expert with the models. But my understanding of the models which were used in the 5th assessment report is that they predominantly included thermal expansion. So to the extent thermal expansion affects pH, those effects would be included. Ice melt is much more difficult to model, and that's been one of the sort of limitations of the global models that have been used in the 5th assessment report, is the general idea that the sea level rise coming out of those models is a conservative estimate because of the additional sea level rise from ice melt.
Okay. The next question is, how much of the coastal acidification is driven by atmospheric CO2? I know that the modeling was done on general ocean acidification, but if you could address that, that'd be great.
So yeah, coastal acidification presents a significant challenge. Even in the Northeast region, there is a lot of regional variability in carbonate chemistry. So you see differences in pH in the Gulf of Maine compared to the Mid-Atlantic. The global models in the 5th assessment report were relatively coarse resolution, and so they're not really designed to capture sort of the regional scale ocean acidification. So work is going on now to develop sort of regional, they're called downscales. You take a global model, link it to a regional model, and downscale the global effects to your region. And so there's work underway now to generate these sort of downscaled, higher resolution regional models, which will be better able to capture regional and coastal acidification, and then also some of the smaller scale changes in current patterns. But that's work that's going on now, and the results were not available for our assessment when we completed it.
Thank you. In New England waters, how are scientists monitoring ocean and coastal acidification? Are there monitoring instruments that are particularly adept at this?
I guess I'll answer again. Yes, there are. So the monitoring has, both components. There's instruments in coastal regions, and there are also instruments and monitoring activities going on at sea. The number of different instruments. You can measure the CO2 in the water, you can measure pH, you can measure alkalinity, and there's several efforts underway to improve the instruments that are available. So there's a lot going on, and it's a very active area for researchers and for technology development.
Great. I'd like to ask a question of Hauke. Where can states and municipalities go to for ocean acidification information relevant to their region?
I think one place in many cases are the regional ocean observing systems around the United States. So in the Northeast, for example, there's some organization called Narragansett that maintains data sets on marine conditions. Local research institutions are a good resource. The fisheries service offices, like the one that John works in, are another good source. And I think there are NOAA web pages dedicated to ocean acidification. So I think it takes a little bit of searching. There's no single place I would direct everyone to, but those are good places to start.
Great, thank you. I have a question for John. There were fish, particularly flatfish, in the high-risk red boxes with some of the shellfish. Can you elaborate on the life history, diet, or other factors that make some fish species more vulnerable to ocean acidification? And I guess I'll expand this if Hauke wants to comment as well. Are there any factors that make different mollusk species also more vulnerable?
Hauke, do you want to answer first for shellfish, and then I can answer for finfish?
Sure. Yeah, with shellfish, some of the variability in response is due to the extent to which the shellfish rely on aragonite versus calcite to build their shells at different points in their life cycle. Different species of shellfish use different biochemical techniques for building their skeletons, another source of variability. And I think that when it comes to other marine species, there's some evidence that acidification affects the way that squid, for example, and I think some finfish as well, are able to build their internal organs, including those responsible for allowing them to navigate and find their way in the water column. And so there's a lot of research on this now, and a lot that we still don't know about it. But I think that in the coming 5 to 10 years, we'll get a much better fix on exactly which species are likely to be affected by this. And John, you may be able to add more there.
Yeah, just a little bit. I agree that the next 5 to 10 years, we will know a lot more about the effects of ocean acidification on finfish, on shellfish, on mollusks, on the crabs and lobsters, and that information will be critical to understand how ocean acidification is going to affect the resource species. From the fish side, as Hauke mentioned, there is this sort of several studies which indicate that ocean acidification may affect fish's ability to smell, and so it's the mechanisms aren't exactly clear, but the change in carbonate chemistry is just it's having a wide range of effects across multiple groups of organisms, affecting calcification, potentially affecting olfaction, affecting physiology. So we're really just beginning to learn what the impacts are going to be.
Thanks. I have a question that I'm going to address to Bärbel. So far as you can tell, because I know it's very difficult to estimate pH through geologic history, but has global ocean pH ever been as low as the projected RCP 8.5, which is approximately pH of 7.5?
7.5 is very low. It's probable that during the Cretaceous it was that low as it's going to be. But as I tried to explain before, the difference between the Cretaceous and today is that the carbon dioxide levels were high for a very long time. So for this weathering of rocks on land, for that effect to kick in, that takes about 20 to 40,000 years. And so I don't know whether you remember that in my figure, the recovery at the PETM actually took somewhere on the order of 70 to 100,000 years from that acidification condition. So that kind of puts it into perspective of how long this is not an effect like the ozone hole or acid rain that we can easily address and reverse. It is something that will stick with us for a very long time. So even though we have seen these low values, they have not actually been put into place at a rate as we are putting it into place today, and that makes the big difference.
Great. So I think this will be the last question. In some reef fish, the adult and young of the first generation exposed are greatly affected by ocean acidification. However, some second generation fish are not affected. Is any work like this being done for higher latitude fish species?
Yes, I know of several projects where this is being done, and this raises an important issue in general about how we think about the effects of climate change on marine species. Just to make the situations more tractable, we oftentimes make the assumption that the biology or ecology of the species is not going to change going forward. But the person who asked the question, identified studies which show that individuals can acclimate to new conditions. So, as when presented by new conditions, their physiology can make changes so that they're better suited for those new conditions. And groups of individuals, populations, can also adapt to new conditions. That's an evolutionary response, and that can happen relatively quickly too. And we don't really have a good idea of what the acclimation capacity is or what the adaptive capacity is for most marine species to ocean acidification, to warming temperature, to changes in stream flow, and that is an area of active research.
Great. Thank you. That's all of our questions for today's webinar. If you have any questions that were not addressed today, you can email them to us at cipinfo@americangeosciences.org, and we'll make sure to send your questions along to the speakers. We will be posting today's recorded webinar on our Critical Issues webinar page soon. You can visit the website to view this webinar as well as previous ones and check out the webinar schedule for upcoming webinars. And again, we have another webinar coming up on March 30th. Thank you very much, Bärbel, John, and Hauke for speaking today. This concludes our webinar for today. Thank you for joining us, and we look forward to seeing you at our next webinar.